Thank everyone to be with us tonight or in the morning on the museum. So this is the very first kickoff seminar for the Chinese New Year. It's absolutely my pleasure to welcome our today's speaker, Dr. Ayin Ling from Princeton. She used to be my colleague, but she's off to Princeton. So I think now it's sounds like a much better choice than NIH. So Ayin currently is a assistant professor in Department of the Molecular Biology in Princeton. So she is a incoming a new professor in Princeton. And I think it's just a little bit about a year or so. So Ayin actually completed her undergrad study in applied biology with biotechnology in Hong Kong Polytechnic University. And then afterwards, she did her master's degree from the University of Hong Kong. Now, as a European Union Marie Curie Fellow, she completed her graduate study at the Pasteur Institute in Paris. There, she studied identify innate lymphocytes precursors from the blood and tissues of healthy individuals. And with the support of a Human Frontier Science Program Fellowship, she joined the lab of Yasmin's lab in NIH for postdoc training, where we actually knew each other and then had a lot of great time. So during her postdoc training, she found a maternal infection can provide pre-birth immune education to the offspring in a tissue-specific manner. And together, her work in both human and the mouse model in mouse immunology demonstrate that the immune system is highly plastic and then can adapt to environmental challenges even before birth, which I think end up with a lot of great work and really truly brilliant publication. So currently, Eileen, as I just mentioned, located in Princeton, and her lab actually mainly interested in establish a mechanistic understanding of how maternal infection impacts offspring's long-term tissue-specific immunity, which I think she's going to talk about today. And her lab's goal is to interrogate how interactions with microbials in the form of pathogen or commensa contribute to the evolution of our immune system with the ultimate goal to improve healthy trajectory in children, and also to explore how does maternal immunity adapt to support the physiological changes during the pregnancy and the lactation. There are different interesting in her lab, which including, but not limited, in exploring the role of maternal helminth infection on offspring tissue immunity, and then deposition to immune disorders. And second part is dissect the impact of the maternal infection on breast milk composition and function. And lastly, but not least, to understand the mechanisms of maternal immune adaptation to pregnancy and lactation. So with all these really marvelous works, she got numerous awards, which including in France, the L'Oreal for Women's in Science National Awards in France, also International Rising Talent Award. She continues to get the NIH Fellow Award for Research Excellence, both 2019 and 2021. 2021, she also got Sidney John Pesca Postgraduate Award for Cytokine Society. And 2022, she got NIH Cytokine Interest Group Bill Paul Awards for the best paper in cytokine research. So I think without further ado, we are very exciting to have you on our platform. Very much looking forward to your talk. Go ahead, Eileen.
Thank you very much, Hans, for such a nice introduction and the opportunities to present the work that we are doing and where we are for moving forwards. First of all, Happy New Year, everyone. I hope you all have a good celebrations and all the best for the coming year. So as Chance mentions, my strongest interest or what really driving my research is really maternal offspring immune crosstalk. I would like to think about such relationship or in actually any kind of relationship as a partnership. both mother and the offspring has to benefit during this critical window. So I think this is really some things that can drive our species, like for the benefit of our species. So I just launched my lab about a years ago. There are two questions that really close to my heart, and I really think about that day to day. And we put all of our efforts in understanding first, how does the maternal exposure, what I mean for maternal exposures, it can mean various kind of environmental exposures that we encounter day to day. For example, different kind of infections, different kind of diets that we take, or even when the mother are traveling, they could encounter new microbes. All of these environmental exposures can activate the mother immune system. And I want to understand, how does this kind of exposure can impact on offspring immune system? I think about this most of the time as an immune education. And so the first direction is really, how does the mother impact on offspring? The second questions that we think about a lot is really adaptation. We think about pregnancy and lactation as a very unique situations for the life of a woman. Fetus is like genetics in identicals to the mother. How does the mother immune system able to adapt to pregnancy and lactations are the second directions that our lab putting a lot of effort in understanding this. So why and how do I come with these questions in mind? So I think I have been always fascinated by the developments of our immune system. I always fascinated by how a single fertilized cell can divide and differentiate and becomes a whole human being. I think this is such a greatest mystery in biology, but this is probably the miracles that all of us have done at the very beginnings of our life. So along with developmental biology, I'm always fascinated by how our immune system, like how a single hematopoietic cell can give rise to diverse immune cell with very distinct function. I think immune system honestly is the best example of diversity that they have. We have so many type of immune cell, but they have to collaborate together to maintain the balance between our body and the environmental challenge. So we all live in an environment that we constantly encounter different kind of microbes, ranging from virus to bacterial. And I think it's a big part of the world. People are still living with parasitic worm. So how does our immune system as a front line to maintain the balance with these microbes? And at the same time, how does these microbes, either in the form of commensal or pathogens, plays a role in shaping our immune system? Is the questions that I want to address for my career. So all of these process, the developments of our immune system start in utero in parallels to all the organogenesis in a highly regulated manner. And immediately after birth, our burial tissues start to colonize by diverse microbes, especially in our skin, our lung, and our gut. So proper developments of our immune system really require proper environmental input in a timely regulated manner. And this can have a long-term impact throughout the adulthood. And once this window is closed, it is very difficult or even impossible to reopen again. So I think a lot of epidemiology studies have suggested that during this critical window, the experience of the mother can shape the offspring immune system, either through placenta transfer or through breastfeeding. But despite all of these clinical or epidemiology studies suggest, we don't actually understand how does the mother impact on offspring immune system. So I begin to think about how does the mother environmental exposures impact on offspring immunity. As I mentioned at the very beginnings of my talk, I'm broadly interested on different kind of infections, diet, and also microbes that encounters by the mother. How does that impact on offspring immune system throughout the adult life? So a few years ago, I had the opportunities to join Yasmin Lab at NIH. We first tackled these questions in infectious disease. So a lot of time, infections during the pregnancy has been focused on those severe or vertical transmittable pathogens, such as Zika, Toxo, or Listeria. And this kind of pathogens can really lead to the catastrophic consequence on the offspring, such as developmental defect or premature birth. But when I think about this, I think a lot of time pregnant women encounter pathogens that are transient or asymptomatic during the pregnancy. For example, it can be a common cold, a stomach flu, or nowadays coronavirus can be asymptomatic as well. So the mother can cope with the infections without direct transmit the pathogens to the offspring, but they still have immune activation during pregnancy and lactation. I start to wonder, how does this transient and asymptomatic infections that encountered by the mother impact on offspring immune system for the long term? So a few years ago, to address these questions, we decided to use a very nice model, Yersinia pseudotuberculosis, to address these questions. So Yersinia pseudotuberculosis is gut pathogens. It can infect human. And what we use is a Yolk M mutant strain of Yersinia. So what you can see from here, we infect the time pregnant mice with Yolk M mutant strain of Yersinia around day 10 of gestation day, which is the end of first trimester. We observe a very minor body weight drop after infections, but they are able to recover and deliver comparable numbers of offspring compared to the non-infected mother. We can track the bacteria by measuring the DNA inside the feces that you can see by day 3 is the peak of infections, and they get clear very quickly. By day 9 post-infections is the times that they're about to deliver the pups. They are completely clear with the infections. So we look for the bacterial disseminations by calculate the colony forming unit. The peak of infections is at day 3, but we rarely see the bacterial disseminations into the circulation. About 20% of the mice have the live bacteria in the liver and in the spleen, but we never detect any live bacteria from placenta and from fetal liver. So we think your mutant strain of Yersinia give us a transient and maternal restricted infections model. What we really want to understand is what is the impact on the offspring. So when these offspring reach adulthood, we look for their burial tissues immunity from skin to the gut to the lung across all the lymphocyte subset. So in the following slide, I'm comparing the offspring delivered by the mother who received PBS, showing as maternal control, or the offspring delivered by the mother who has been previously infected. So what we found from that study at the very beginning is we found the offspring delivered by the previously infected mother have the inductions of TS17s in their GI tract that you can see from here. They express a high level of rho gamma T. And this is both in frequency and in cell number in the small intestines. We see such inductions also in the large intestines, but not in the skin nor in the lung. And we don't observe other alterations across all the other lymphocytes that we check. So I start to wonder, how does these transient infections experienced by the mother can have a long-term impact in the gut-specific immunity? When does it happen? Is this something that happens before or after birth? So we did a cross-fostering experiments that we took the offspring delivered by the previously infected mother from day 1 post-birth, and we give it to a naive non-infected mother. And vice versa, we took the offspring delivered by the PBS-treated mother from day 1 post-birth, and we give it to the previously infected mother. And really, what to our surprise, we found that so here as what I have shown, if the offspring are delivered by the infected and fostered by the same mother, they have the inductions of TS17s in their GI tract. But what we found is no matter who was fostering them, as long as they are delivered by the infected mother, they have the inductions of TS17 cell in their GI tract. And what it means is transient maternal infections can imprint the offspring gut TS17 cell prenatally. So we have a series of hypotheses to explain this phenomenon. And one of that is through the serum soluble factor can pass through the placenta. And to test this hypothesis, we decided to do a serum transfer experiment. So we took the serum from the infected mother from day 3 and day 5 post-infections, and we give this serum to a naive non-infected mother at gestation's day 13.5 and 15.5. And what we found is by simply transferring serum itself, the offspring delivered by the YOP-M serum recipient also have the inductions of TS17s in their GI tract as the comparable numbers to the offspring delivered by the infected mother. What it means is the soluble factor from the infected mother are able to induce the offspring intestinal TS17 cell. So we decided to look what is increased in the serum. And as immunologists, the first things that we look for are the cytokine. So we screened through a series of cytokine, and we found particularly IL-6, interferon gamma, GCSF, and TNF alpha are increased after infections. So honestly, these cytokines are not a surprise to us because we know this is the cytokines that obviously will increase after YOP-M Yersinia infections. And to test any of these cytokines from the serum of the mother can impact on offspring gut immunity, we decided to do a gain of function experiment. So we injected time pregnant mice at gestation 13.5 with this cytokine. And we found by injecting IL-6 alone is sufficient to increase TS17 cell in their GI tract. And when we block IL-6 prior and during infections, it completely dampened the TS17 cell in the GI tract of the offspring. So what it's telling us is IL-6 is not only sufficient, but it is also essential to promote the offspring TS17 cell in their GI tract. So now we found the factor IL-6 can drive the tissue's immunities in the offspring. So we decided to look how does the IL-6 are able to do that. So I start to look for the fetal IL-6 receptor expressions inside the gut. And to our surprise, we found indeed the fetal gut epithelium cell, the EpCAM positive cell, they express intermediates of IL-6 receptor that we see that by flow cytometry and confocal imagings. And more importantly, the downstream signaling of IL-6 is phosphostatin. We found the offspring delivered by the previously infected mother or the ones that inject us with IL-6, they have the inductions of phosphostatin in their intestinal epithelial cell. So what it means is the the gut epithelial cell are able to respond to the environmental exposures of the mother. To rule out IL-6 receptor expressions by the gut epitheliums are really the ones that contributes to the inductions of TS17 cell in the GI tract of offspring. We decided to specifically delete IL-6 receptors in the gut epitheliums of offspring. And I hope you can appreciate from here. So to do this, we actually cross the villi Cree and IL-6 receptor flux mice to each other. So in our breeding scheme, we have the mother express IL-6 receptor intact, but when we cross it to the villi Cree father, the mothers deliver IL-6 receptors specific knockout in the epithelial cell or the ones that intact. So you can see from here that the Cree positive littermates, they actually have a specific knockout in the gut intestinal epithelium as compared to the littermates control. And when we inject the mothers with IL-6 during the pregnancy, what we found from here is the reductions of TS17 cell in the GI tract if we delete the IL-6 receptors in the intestinal epithelial cell, suggesting that IL-6 receptor expressions in the gut epitheliums of the fetal are really essential to confer this long-term immunity. So far we found IL-6 is an important factor from the mother that can imprint the fetal gut epithelium that lead to the inductions of TS17 in the GI tract of offspring. But there was still really a puzzling factor. How does early life exposure with one cytokine can have such a long-term imprinting in the immune system in the offspring throughout their adulthood? So when I was really puzzling with this, I read a beautiful paper from Kim Jensen. So he's a developmental biologist specialized in the gut, and he put out a very beautiful paper that he used series of lineage tracing approach and found all the fetal epithelium cell, regardless they express LGR5 or not, they will become the LGR5 positive intestinal epithelial stem cell when they reach adulthood. So after reading this paper, I was really excited because what in my mind is if the fetal intestinal epithelial cell are express IL-6 receptor and they have been activated during fetal life, they might have a long-term imprinting in the intestinal epithelial stem cell pool. So after reading this paper, we decided to get the LGR5 reporter mice, which allow us to sort the intestinal epithelial stem cell from the offspring delivered by the control or the mother who has been injected with IL-6 during the pregnancy. Because a series of papers have shown the intestinal epithelial stem cell, not only in the intestines, but also across various tissues such as skin and in the lung. The epithelial stem cell can actually carry epigenetics memory from previous insult. Based on this, we decided to look for the chromatin accessibility profile of the offspring delivered by the maternal control or IL-6 injected mother. So what we found when we do ataxic, what we found is the chromatin accessibility was increased in the intestinal epithelial stem cell of the offspring delivered by the IL-6 injected mother, with around 1,000 regions open in the IL-6 injected mother, as opposed to only 71 regions opens in the control offspring. So what it means is there's more open chromatin accessibility in the offspring if the mother has been injected with IL-6 during the pregnancy. And we know intestinal epithelial stem cell are the ones that give rise to all the diverse epithelial cell inside the gut. So what it means to have more open chromatin accessibility, we start to wonder whether it changed the function of the intestinal epithelial cell or whether they changed the intestinal epithelial cell subset. To test this, we decided to do a single-cell RNA-seq that will allow us to look for different subset of intestinal epithelial cell, and on top, we can look for the transcriptomic profile of each subset. So when we do the single-cell RNA-seq from the offspring delivered by the maternal control, or the offspring delivered by the mother who has been injected with IL-6, we did not observe any striking difference across all the subset of the intestinal epithelial cell. However, when we compare the transcriptomic profile of each subset, really to our surprise, we found the stem cell and the anterior side. They have much higher MHC class II machinery and also bacterial defense profile that you can see from here. So bacterial defense machinery profile, which makes complete sense, it suggests that the offspring might able to have a better protections against infections. But for MHC class II expressions in the stem cell, they are actually a recent paper that show MHC class II expressions in the gut epithelial cell are very important or correlates with colitis or gut inflammations. Based on this study, we decided to look how does the offspring respond to different kind of gut infections and inflammations. So to do this, we decided to infect the offspring with Salmonella. And we took the offspring delivered by PBS or IL-6 during the pregnancy, and they reach adulthood, we infect them with Salmonella. And what we found is the offspring delivered by the mother who has been injected with IL-6 are able to control the bacterial burdens better, that they have the reductions of bacterial burdens inside their feces. They have reduced bacterial disseminations to the circulations. And more importantly, they actually survive battles after this virulent strain of Salmonella infections. On the other hand, we also want to understand, other than infections, how does the offspring respond to inflammations? So we took the offspring delivered by the control, YOM infected or IL-6 injected dams, and we induced gut inflammations by putting DSS in their drinking water. And when we look for the parameters of DSS-induced colitis, we found the offspring delivered by the YOM infected or IL-6 exposures mother have much more severe body weight drop at the chronic phase of in this DSS-induced colitis, and they have also much shortened length of colons, which suggests that they are much more severe in this DSS-induced colitis. We also test this in another model, which we use naive CD4 T cell induced colitis model, and we found the offspring delivered by the YOM infected or IL-6 injected mother are much more severe in both models of colitis. So collectively, what we found is a transient infections that experienced by the mother during the pregnancy can increase IL-6, and this IL-6 can activate the fetal intestinal epithelial cell, which can lead to epigenetics imprinting in their stem cell pool. So this can allow the offspring, when they reach adulthood, they have heightened immune response. What it means to have heightened immune response is they can respond more to the microbiota. So they have accumulations of Th17s in their GI tract. And this can come with the protections against infections, but this also allow them to have a higher predispositions to inflammations. So altogether, I put forward ideas about maternal infections driven immune education models. What I think about this is the maternal encounter during the pregnancy can be co-opted by the offspring to develop optimal immune fitness. What I mean for this is if the mother encounters infections during the pregnancy, they might think about their offspring are going to enter an environment that they will have more infections in their life. So they want to prepare the offspring to fight against infections. But I also oftentimes think life is a balance. When you have a protections against infections, you might come with a consequence to have a higher susceptibility to inflammations. So this is the ideas that I come up with at the end of the works that I have done during my postdoc. And if I think about that, I think other than the form of infections, we also all live with different kind of multitudes of microbes in our walls. And most of the time, this is the forms of commensal that without causing any pathogenic impacts on our body. But nowadays, with urban living, sanitizations, antibiotics, or deworming treatments, this has already significantly shaped our relationship with the commensal microbes. At the same time, a series of epidemiology data have shown an alarming increase of immune disorder, such as allergy and autoimmune disease in many high-income society over the last few years. So I think immunologists explain this as hygiene hypothesis that we think the reductions of our relationship with the microbes can lead to immune dysfunctions. And so we start to respond to the self or harmless agents. And amongst these disappearing microbes, one of the partners that really attract my interest is helminths, or generally known as parasitic worms. I think this is really one of them that we have been evolved for a long time, but has been recently removed from our body, especially humans in many high-income societies. So I think what is unique about humans is it can actually induce regulatory and tissues repair immunity. And so these can dampen the inflammations. At the same time, this allowed the humans to live inside our bodies for the long term. So I really think our relationship with humans is symbiotic. So then the questions that I have is, if such a partnership are so essential in our body, and now we remove them, is that the reasons that we start to see a lot of immune disorder in our society? And the works that I have done also shows the early life educations are very important. So the questions that I want to address is the maternal-driven hygiene hypothesis. So the hypothesis that I have is the maternal-Helminth's partnership might produce an essential immune education spectrum to optimize the immune educations in the offspring. And if we remove such important factors during critical developmental window, it might lead to immune dysfunctions. And so the generations have more immune disorder. So this is the project's lead by a very brave postdoc, Chris, coming all the way from Brazil to join my lab as a founding members. So we think about this a lot. And I think one thing that I really want to highlight is this project is really not to study, is not to really reinfect the humans populations with helminths. The goal that we have for this project is we want to identify what is the factor derived by the maternal-helminths partnership, and whether we can use this factor to optimize the offspring immune system without reintroduce helminths to our humans population. So the first projects that we are trying to address is the pre-birth hygiene hypothesis. And the second directions that we put a lot of efforts doing in the lab is immune adaptations during pregnancy and lactations. So I think this is crystal clear that pregnant and lactating women go through a series of physiological alterations during pregnancy and lactations in order, which is necessary to actually adapt and nurture the fetus and the newborn. So from the immunology perspective, there's a lot of epidemiology study have shown the serum cytokine actually change during pregnancy and lactation. And I think one very obvious example is some autoimmune disease patients, they always experience reduced disease severity during pregnancy, and this relapse after lactations. And obviously, immune adaptations during pregnancy and lactations are essentials. And when maladaptations occur, that can lead to pregnancy complications and preterm birth. But when I think about this, I think a lot of our understanding for immune adaptation has been focused on the circulations for the very understandable reasons, because blood is really a place that easy to get sample. Or the other side that has been a strong focus is in the placenta. And this is also because this is the tissues that so easy to get after delivery. But when I think about immune system, I always think about our immune system functions at the burial tissues, such as gut, skin, and lung. So the other directions our lab are working on is to try to understand the maternal tissue's immunity across these burial tissues. How does the tissue's immunity adapt during pregnancy and lactation? And this project is leading by Chen Yan, my first graduate students, and Jojo, who joins the lab very recently. So this is very preliminary, and I hope that in the near future, we can share more data about these two directions from the lab. So before the end, I would like to thank the founding members from my lab, Chen Yan, Chris, and Jojo, who are very brave and jump in to do these projects together with me and have a strong belief in me. So it has been a great fun to work together with them. I also want to thank the funding, Bronco Wise, which is my transitions fund, and also Rutgers Cancer Institute of New Jersey that allow us to do some pilot experiments in cancer. So with this, I'm very happy to discuss more about the idea in maternal offspring immune crosstalk or answer any questions that you have in these directions. Thank you very much for coming to my talk early in the mornings or late in the evening from every part of the world. I'm happy to take any questions that you have. Thank you.
Well, thank you, Ayin. Well, I think this is probably the first time I actually hear your entire talk. Now I can appreciate it more.
Okay.
So, anyone in the audience have questions, please just raise your hands or put your question in the chat box. Charlie, go ahead.
Oh, hey, thank you so much for a really beautiful talk, and I think it's eye-opening for me personally, because I haven't really appreciated the importance of this maternal immune education. So I guess my question is probably pretty general. So if we do this immune education during the pregnant stage as compared to after post-birth, is there any advantage or disadvantage in terms of, let's say, the immunity acquired or the immune response?
So how tight, I guess the questions come as how tight is the window for us to educate the offspring immune system, right? I think one thing that I really want to highlight here is the works that I present is really the very beginnings of this field. We only work on the gut, but I think there's a lot of other tissues to explore. We only start with one pathogen, Yersinia, and we identified IL-6. And there's a lot of other pathogens that I think they work differently, and they might educate the offspring immune system in a very different way. We found IL-6 receptor expressed by the gut epithelium. This also opened the door because all of the other tissues they might express all the other cytokine receptor at different way. For example, skin or lung, they might express other set of cytokine receptor. And so the questions, I think I don't have a clear answer for your questions. I think in general, my view or my speculations for this is there's probably different ways to educate the immune system, but this might be very depends on the cytokine or very depends on the pathogens that we encounter. So let's say for the case of our model through IL-6, we have done that during the pregnancy. It's a pity that we actually didn't follow up with the other window. But my speculations for that is because the work from Kim Jensen, he actually showed by lineage tracing, before 2 weeks of life, all the epithelial cell can become stem cell. So if these models work, I think my speculation is from pregnancy and until post-birth, 2 weeks of life, there's a windows of educations with IL-6. But I think for all the other cytokine, it might work in different way. Am I answer your questions?
Yes, I think that's great. The other may be a really wild idea. So because I mean for the newborns we give like all different kinds of vaccines, right? Will there be any advantage if you give the vaccine during the maternal still I think in the maternal stage?
Yes, I think certainly I think maternal vaccinations, I think usually by third trimester, pregnant women indeed receive several rounds of vaccinations for the purpose of protect the offspring.
I see.
A lot of that are actually true antibody, right? Because they want the antibody to transfer across the placenta. We want the antibody transfer across the breast milk. But I think I view vaccinations probably like a transient or minor infections, right? So it could also induce a series of other cytokines in addition to antibody.
Yes. I think this has to be very elegantly balanced between the immune response and the damage.
Yeah. So yeah, is there complicated? Let's say is there complicated partnership, but I think it's still very important to understand the details and at the mechanistic level, so we can really utilize our knowledge in these areas to improve women's and children's health.
Absolutely. Thank you so much.
Of course.
Okay, so next question from Harris Han. Thanks for the great talk. Were the infant in a germ-free environment so that we can make sure that IL-6 are strictly coming from the mother?
Yes, I guess the question is whether, so we actually have done these experiments with the germ-free mice. So at some point we are wondering whether microbiota is really essential in this process. We have done this, we inject IL-6 to the germ-free pregnant mother, and we analyze the offspring. So what we found from that experiment is if the offspring, the IL-6 injected offspring stay in germ-free, they don't have the inductions of Th17. But if we conventionalize the offspring actually legitimate to our SPF environments, we start to see the inductions of Th17. So when we think about these experiments, we see that is there are two signals are required to induce Th17 cell. One is the IL-6 exposures from the mother and activate the fetal intestinal epithelial cell. And the second is they need another hit of microbiota. So they need two signals to increase the Th17 cell. Now, there's one other experiments that unfortunately that I wasn't able to do is I want to see the epigenetics imprinting, whether it's require the microbiota colonizations. So the ideal experiments for that is using LGR5 from the germ-free and expose that to IL-6. But because we don't have the strain in our germ-free facility. So that was one of the dream experiments that I haven't done. So to answer your questions, we have done that. We think microbiota is another important signal. Am I answering your questions?
Okay. Well, I mean, I think if audience, well, we'll see. I mean, if there any response. The following question, I think the next question from Zhang Yuxia. So, very nice talk. What do you think will happen if there is repeated infection? Do you see maternal infection with risk of food allergic or IBD? And what happen if offspring experience further infection?
Yes, I think our model, so we only do one model, right? Yersinia with IL-6, and this is a really transient infections, and they are very minor. So I think I think I don't have a clear answer for you, but my speculation is they must need a very tightly regulated infections because infections is a cost for the mother. And how does the mother balance herself to recover from infections without losing the offspring? We haven't done this kind of experiments to see repeat infections, or even if the mother first have a stomach flu with a gut pathogens, and if we come back with influenza virus infections, how does that impact on the offspring? I think all of these are very good questions, and we can actually address that in our mouse model. Do you think that increase the risk of food allergy and IBD? I think at least from these models suggest that it increased the risk for inflammatory bowel disease. But whether that will be further increased or there's a threshold for that, I think all of this can be addressed in the mouse model.
Okay, so next question from YUPC. What level of IL-6 in other fetal tissues, such as amniotic fluid and fetal liver cells, and IL-6 receptor in other fetal tissues?
So this is a very good question. We actually did a careful study because at some point I was really unsatisfied why this go through the serum. In theory, we actually see that in the fetal serum. So I spent a lot of time collect the fetal serum, and we see the inductions of IL-6 as well. I was very puzzling why this is really gut specific. So unfortunately, we didn't look for the fetal liver. We looked for other burial tissues, including fetal skin and fetal lung, and of course, fetal gut. So we found the IL-6 receptor is actually specifically expressed by the fetal gut epithelium, but not from the skin and from the lung. So I think this IL-6 receptor specificity expressed by the fetal gut explain why explaining the tissue specific imprinting. But I think this is another directions that I keep thinking all the time is what is the other set of cytokine receptor can be expressed by all the other fetal tissue. And we are diving all the single cell RNA-seq data that available that has been published to look how is the cytokine receptor expressed across all the fetal tissues.
Okay. So next question from Chen Xiongwen. Great talk. Is the maternal antibody transferred to the fetus during pregnancy in mice? If so, is it related to the establishment of Th17 cells in the GI system?
Certainly, maternal antibodies can transfer across the placenta. I think the major class of antibody get transferred across placenta. It should be IgG. Whether that is the ones that induce Th17 cell, we haven't tested these directions. I think I can think about the good experiments to do for this is we can probably use mu-MT knockout mice to see without antibody whether we still have the Th17 inductions. I'm sorry that I don't have a good answer for this, but we haven't tested that yet.
Next question from Soon Kwang Chung. So very interesting talk. In relation to your first bullet point on how maternal exposure educates offspring immunity, how do you think placenta plays a role in this, and how important are the placenta immune cells like Hofbauer cells. I don't know what's the cells contributing to offspring immunity.
I guess our view for placenta, it's probably to create a barriers between the mother and the fetal, and also to provide the essential nutrients. How does the placenta control what is being transferred or not? I guess that for sure play a very important barriers between for the signals to transfer across, right? I don't think I have a good answer for these questions, but certainly this is some things to think about further.
Okay, Tone, are you there?
Yes.
Yeah, go ahead.
Hi, Eileen. Yeah, this is Tony. Yeah, from the CE Education team. Yeah, I managed the B cell. So, but I want to ask a question about the T cell. So, based on what I learned from the textbook, the IL-6 together with the TR-beta that can promote the Th17 differentiation while inhibit this Treg, right? So I kind of wondering, for your model, you propose that the IL-6 work on with the IL-6 receptor on the intestine and epithelial cells. Do you think the IL-6, the increase this Th17 in the offspring, like in the gut, can be T cell like intrinsic, or you think this IL-6 need to go through the intestine and epithelial cell first, then affect on the Th17? And also in a model, typically people see like a antagonistic relationship between Th17 and the Treg. So in your model, in a Yersinia infection or IL-6 administration, you see an increase of Th17. Do you see a decrease in Treg?
Yes, both are very good questions. And let me first answer the second question, is whether we see changes in Treg. We actually did see that, but that is more minor, is not as striking as the inductions of Th17 cell. And the first question is whether this is T cell dependent or not, or it has to go through the intestinal epithelial cell. I think this is not T cell dependent for the reasons that we have inject IL-6 in the rat knockout mice, which are deficient in T cell, and we still see a protections against colitis when we transfer like in the nice CD4 T cell transfer model. So I think this IL-6 can educate so I think in our models, the IL-6 is really working on the intestinal epithelial cell, but not on the T cell. So, but I think for sure they are actually work coming from Junhyu. I think he used more poly-IC as a methanol activations model. And in his model, that is actually T cell dependent protections against infections and also susceptibility to inflammation. So to answer your questions, I think for short, our model is more relying on the epithelium, but there are some other situations that can be T cell dependence for the immune education. Does it make sense?
Yeah, that's great. Yeah, thank you. That were very clear answers. So, and also in your summer experiment, you inject the Yersinia like infected maternal serum into the naive mice. So how much serum did you inject? It was very surprising. You can, that amount is enough to drive your phenotype. Yeah.
It was actually also surprised that I don't think that experiments will work, to be honest. I think we inject several rounds, so every other days with like 200 microliter of the serum. And we actually, at that time, there's also already of IL-6 post-infection.
Okay, great. Okay. So if other infection also can induce IL-6 upregulation, even the COVID, right? COVID, some people have the cytokine storm such as IL-6. So do you think that will have similar like impact on your fetal intestine? Yeah,
I think we think about that. We discussed about this a lot actually, whether this is really Yersinia specific or this can be broadly for other infections. I couldn't agree more with you that all the other infections, IL-6 for sure is one of the candidates upregulated post-infection, right? And also from a lot of other inflammations, also chronic inflammations always have high level of IL-6. Do they always come with a benefit on the offspring? I think this is another directions that we have to really study carefully, because I think in our model, this is really a transient infections and it's up regulated for IL-6 and then it go down quickly. I think the things that we haven't addressed is what is the tonic level of IL-6, how much IL-6 we need, because I think at different infections, IL-6 might come with different level. And with COVID, it's not only IL-6, like interferons all will be up as well, right? So how does we balance different kind of different cytokine in offspring immune educations? I think all of these are all very interesting questions, but we just don't have a very good answer for that.
Okay, thank you so much. Hopefully, when you come back to Hong Kong for the home visit, you can also visit the Shenzhen. Yeah, very close by. Yeah, thank you.
Yes, I would love to.
Well, next question, Baochun. Baochun, go ahead.
Hi, yes, I'm Baochun. Very nice talk. So I have a question. You mentioned that the IL-6 induce class 2 in the intestine epithelial cell.
Yeah,
so
I have the same question.
So I wonder what's the role of MHC class 2? And a following up question, whether the class 2 expression is direct or indirect effect of IL-6.
Yes, I think we thought about that a lot, and in recent years, there are actually couples of papers come out for showing the roles of MHC class 2 in the gut epithelium. Do they really present antigen, or do they have other non-canonical roles? I think that's probably, there was one paper from Banajabli published in Nature a few months ago. They used MHC class to knock out mice crossed with Vilicri, and they actually very elegantly show this can, if I don't want to say something incorrect, but I think she was showing this is very important for controlling cancer. But there was a very nice paper published in Nature from Banajabli showing the roles of MHC class 2 in the gut epithelium. And for your questions, what is inducing that? Whether that is solely by IL-6, at least in our models, we see IL-6 injected from the mother increase that. But the works from many other, they actually also show microbiota colonizations can induce MHC class 2 as well. So we don't have a clear answer what is the upstream of MHC class 2, but this is something that I guess as immunologists, we are very interested in.
So Okay. I mean, I probably you have the system because you have the IL-6 receptor, IL-6 receptor knockout. In the knockout mice, whether you're able to see class 2, that will address that question. Yeah.
Yes, that is very easily to be done. Actually, you're right.
Thank you. Yeah.
Well, Aline, I have a full page of questions. So, I think I want to follow up with Baochun's question. So my question is, if you do the same infection in the Vilicri MHC class 2 flox mice, what happened to the Th17 cells?
Oh, if we do the infections, we have done this
because you mentioned that obviously the infection or IL-6 signaling to induce Th17 is mediated by or it's controlled by the epithelial MHC class 2 expression. But if that's causality established, then if you knock out MHC class 2, then the IL-6 injection or any kind of infection shouldn't induce any Th17 cells.
Yes,
My question is, have you done this experiment?
We have done these experiments and we saw that it's gone. I think we have put them in the publications also.
Okay. But then my next question is, so there's a paper from Aviv Rege's lab to profiling the small intestine epithelial cells.
Yeah.
And then the MHC class 2 expression obviously have different patterns based on the specific locations, spatial location, and then subset.
Mm.
And then in her paper that the MHC class 2 dramatically increase in the intestinal stem cells,
Yep.
which actually important for the Treg induction.
Yep.
My question is, I'm curious why in your system, the Treg don't actually plays a dominant role, and
Yes.
then PTH17 cells.
Yes, I remember that paper. I think it's a Cell paper a few years ago, right? I think it's for our model, indeed, we still don't know how is the TH17s get induced. We think it depends on the microbiota colonizations, whether this is true MSC class 2 that yes, have we addressed that?
You can't remember?
I have to think about that carefully. But I think for that paper, they are doing that actually in the culture model, if I remember correctly.
They did both. They did the both. I mean, I'm on the paper, so I know.
Oh.
So it's both on the culture and the in vivo. So I'm not saying it's 100% same as what you did, but I'm just saying that the MHC class to express on the stem cell seems like more dominantly control Treg differentiation. I mean, obviously, this is not some pathological condition or infection. They're doing this in a steady state. So that might make the difference. But I'm just actually curious that to see what do you think about it.
Yeah, I have to think a bit more about this, but we certainly don't. For our model is really inductions of TH17 cell, where the microbiota contributes to the difference between the TH17 cell that we see and the Treg that they see in their model. I think that can be something that to think deeper about this.
Then my next question is, have you actually checked the kinetics of IL-6 receptor expressed on the intestinal epithelial cells?
Yes, we have done. We actually didn't check the kinetics. So we have done that in the fetal stage from day 13.5, 17, and another time point at day 17.5. But we haven't done that postnatally.
But that's during the examination, the IL-6 receptor expressed always high.
Say it again.
I mean, when you do the kinetics, the expression of IL-6 receptor on the epithelial cells always high.
Yeah, it was always high in the fetal stage. But I guess the questions from you is whether it's go down postnatally, right? Whether there's only a period of time. I think these experiments can be easily done. And I wouldn't be surprised that there's a window for IL-6 receptor expression because a lot of receptors are working this way. For example, toll-like receptor 5 is only expressed by the first few days of life in mice. So
the adult mice don't have IL-6 receptor on the epithelial cells?
We haven't checked that yet.
Because if they do, then when you inject IL-6, they should influence both the parent, the mother, and then the fetus.
Yes, definitely. That could be the possibility. But we haven't checked the adult mice yet.
So I think my next question is, so do you know what's actually induced IL-6 receptor? Because if there's a window, then there might be upstream controller for the IL-6 receptor. I mean, for its down regulate and up regulate. I mean, in different window time. That will be crucial because it will determine when you should apply your reagent or infection or whatever you want to apply.
Yes, I don't know what is the upstream of IL-6 receptor, but I could look it up.
I guess my last question is, why this is small intestine? I mean, have you checked the colon?
Yes, we have done that. It's also so we haven't looked for the receptor expressions in the colons, but we have done the TS17 cell. It's also induced in the colon part.
But then, I mean, I just had this because we just did discussing about this kinetics. If the IL-6 receptor have this upregulation and downregulation, which means the MHC class 2 will be somehow induced at early stage of fetus, and then the later stage of the T cell induction is very much depends on the environment shaping. Which means if you have MHC class 2 and then the the pregnant or the fetus exposed to different animal facility, then you probably have different consequences. Instead of Th17 cells, you probably have the Treg cells, or maybe even the other outcome.
Oh, definitely that could be another possibility, because then I think our germ-free experiment would be very nice to address these questions. Meaning, if we conventionalize the offspring to different facility, or we colonize the offspring with different microbiota, whether we see a shift in the immune compositions. My guess is probably likely to be in that way, because what we think about this is the heightened immunity against tampons, right? Yeah.
Right.
Yeah.
And then beyond that, when you just mentioned the rag mice, because these mice obviously don't have the, CD4 T cells. However, these mice actually bring the natural intraepithelial lymphocytes, the IELs. And then the MHC class 2 also can shape the barrier function of that. So my point is, if you're using the rag mice and then to see whether the IEL actually can be modulated by the MHC class 2 in this scenario, whatever you set up for your maternal education system. I'm actually curious to see what's the outcome of these IELs.
Yeah, I think we actually, so for that, we only do that as we do this CD4 T cell transfer colitis. I guess to answer your questions, we can actually inject the mother with the IL-6, and then without any CD4 T cell transfer, we look for their gut epithelium to see MHC class 2 expression. Yes.
Yeah, because obviously, all your exam based on what you just described is the lymphocyte subset. But I think in terms of functionality of burial integrity, it probably have make difference if you have higher MHC class 2 or lower MHC class 2.
Definitely, yeah. There will be all very good experiments to do.
And I think one thing you probably also should think about is, the MHC class 2 also carry the circadian expression level. I'm actually highly suspect the IL-6 receptor also have this.
Oh, circadian for the IL-6.
Yeah, because when you induce the infection or when you induce the cytokine, because the day-night difference. This is something my lab is doing. So I'm not particularly doing maternal immune education, but we are looking at circadian expression in the burial surface regarding the barrier function. So I think this is something probably worth to add one extra dimension to your maternal immune education system. So worth to think about.
Yep.
Yep.
All of these are very good idea. I should think more about that, and maybe some things for the future project.
Yeah, sure. We can collaborate.
Yes, I would love to.
Okay, cool. So I think if I do not see any further questions, I think this is, it's been fun. So thank you so much, Ai-Ying, to share the exciting program and all your study with us. And I think if I don't see any audience have questions, I'm sure Ai-Ying be happy to take questions by email. And if anyone interested in applying Ai-Ying's lab, she's a cool boss, so you should all try to check her lab out. And then I think she's also just opened the lab, so I think anyone want to interested in her work, just go ahead to apply. So with that, thank you so much, Ai-Ying. And then hopefully I talk to you soon in person.
Yes,
so thank you.
Hopefully.
Yeah, thank you, everyone. Really appreciated your talk, Ai-Ying. So hopefully see you soon.
Yes, thank you, Chuan.
Yeah,
Thank
sure,
you.
no problem.
See you, Ai-Ying. Bye.
Thank you very much, everyone.